Hi, my name is Hallie Nienhaus, and I'm here to talk to you today about how the Great Lakes interdunal wetland habitat imposes resistance to gene flow on three aquatic insect species of differing dispersal ability. To start out, how do spatially and temporally heterogeneous habitats affect genetic connectivity among species populations? By spatially heterogeneous, we mean a non-continuous or fragmented habitat, and by temporally heterogeneous, we mean a habitat that shifts and changes through time. Both of these heterogeneities influence the genetic connectivity of organisms across a habitat. They can provide barriers to gene flow, produce isolation by distance patterns, and encourage processes such as local adaptation. Understanding how these habitats affect genetic connectivity can provide a deeper conceptual understanding of how populations of organisms interact with the landscape and can also provide land managers with critical information for the maintenance of gene flow across a habitat to prevent processes such as inbreeding depression and local extinction events. With these heterogeneities in mind, we investigated a relatively understudied and incredibly dynamic habitat known as interdunal wetlands. These wetlands are nested within dune complexes along the eastern coastline of Lake Michigan. They are naturally fragmented on a local and regional scale and are also very temporally dynamic. The middle photo at the bottom depicts fragmentation within Ludington State Park. However, tracts of coastline that either lack a natural dune complex or are developed also contribute to breaks in this habitat. Their hydro periods fluctuate, so during summer months, solar radiation and heat can dry out the wetlands, and then fall rainfall can replenish them. Additionally, on a longer time scale, for example, over the course of a decade, the size and number of interdunal wetlands may vary due to their tie to Great Lakes water levels. Wind can shift the sand dune complexes along the coastline, which can thereby shift the locations of interdunal wetlands across the landscape. Due to the fluctuating hydro period and lack of surface water connection to the lake, these wetlands are actually fishless. However, this does not mean they're void of life. They host very diverse and abundant communities of aquatic insects and also support amphibians, reptiles, and birds. Lake Michigan's eastern coastline, specifically within Ludington State Park, maintains the highest concentration of interdunal wetlands in the world. So here we have an incredible opportunity to study aquatic insect population genetics within this habitat. So as I previously mentioned, interdunal wetlands host abundant aquatic insect communities. And aquatic insects are particularly interesting candidates for population genetic studies because they interact with both aquatic and terrestrial habitats during different life stages, and they also vary in their ability to disperse across the landscape. In this study, we chose 3 species of aquatic insects with different life histories and dispersal capabilities. For our high disperser, we chose Anax junius, otherwise known as the common green darner dragonfly. This dragonfly is terrestrially active as an adult, and they are very strong flyers, with some populations identified as migratory, while others employ diapause or a period of suspended development when habitat conditions become unviable. They also have a large species range ranging from Canada down to Central America. For our intermediate disperser, we chose Notonecta undulata, also known as the grousewing backswimmer. This is a semi-aquatic and flight-capable hemipteran that can complete its entire life cycle within a single aquatic habitat, and relocation to another aquatic habitat typically indicates plastic dispersal as a response to pressures such as predation. Lastly, for our low disperser, we chose Sinus amica, otherwise known as the angler's curse mayfly. And this is a very small lentic-dwelling mayfly with a very short terrestrial adult life stage of around 14 hours. And this 14-hour adult life stage is almost entirely dedicated to reproduction. So this doesn't leave a whole lot of time for this insect to disperse across the landscape if a habitat becomes temporarily unviable. So for this study, we set out to answer 3 general questions. First, does organismal dispersal ability of aquatic insects that dwell in interdunal wetlands determine the degree of population structuring across the landscape? In other words, do organisms that are capable of high dispersal reflect a low degree of population genetic structure, while insects that are capable of limited dispersal reflect a high degree of population genetic structure along the coastline? Additionally, how do gene flow and genetic drift interact at the regional scale? Does the strength of isolation by distance increase with decreasing dispersal ability? And lastly, do differences in environmental conditions along the coastline correlate to differences in genetic diversity within insect populations? Based on dispersal ability of our insects, we hypothesized that for our low disperser, our mayfly, we would observe relatively high genetic distance with increasing geographic distance or at all scales of geographic distance. With our intermediate disperser, we hypothesized to observe increasing genetic distance with increasing geographic distance. And for our highly dispersive dragonfly, we predicted that we would see relatively low genetic distance among some populations at all scales of geographic distance. Our study sites are all situated along the eastern coastline of Lake Michigan and range over 4 degrees of latitude. Starting at our most northern site, we have Sleeping Bear Dunes National Lakeshore, and then moving south, we have Ludington State Park, Saugatuck Harbor Natural Area, Warren Dunes State Park, and Indiana Dunes National Lakeshore. We collected approximately 10 target organisms per species per focal sampling site, and we sampled our ponds from April to October on a monthly basis. We stored all of our insect tissue in genetic grade ethanol to prevent degradation of DNA, and then we collected available at-site environmental data, including water temperature, dissolved oxygen, geographic coordinates, pond depth, and pond area. We also collected data on the total available wetland area of wetlands at each site using aerial imagery in ArcGIS. For our genetic methodology, we started out by extracting DNA from each sample and amplified the CO1 mitochondrial gene to barcode all of our samples and confirm species ID. After confirming species ID through barcoding, we used restriction enzyme associated DNA or RAD sequencing to collect genomic single nucleotide polymorphism data from confirmed species samples. With this RAD sequencing data, we quality filtered all of our reads, removed PCR clones, demultiplexed our reads, de novo mapped our sequences, and then called single nucleotide polymorphisms to calculate population statistics, all using STAX version 2.0, the bioinformatics pipeline. We then tested for isolation by distance, population clustering, and correlations between genetic diversity metrics and environmental data for all species. Interestingly, our barcoding results revealed some unfortunate but intriguing results. For our mayfly species, we thought we had a continuously distributed species along the entire coastline, and this was based on identifying these incredibly small mayfly species in the lab under a microscope using morphological characteristics. However, barcoding told us a different story. In this case, we had two morphologically similar and closely related species of mayfly along the coastline. One species, Canis amica, we found at our 3 southern focal sites, and then Canis diminuta we found only at our 2 northern sites. Although this was unfortunate for the incorporation of a low disperser into a population genetics data set, we found this incredibly interesting as the species distribution seems to align with Michigan's forestic line of tension, where essentially the northern portion of Michigan is dominated by coniferous vegetation, while the lower half is mainly temperate deciduous. Based on this information, this leads us to believe that Canis amica is restricted to our southern sites and Canis diminuta is restricted to northern sites. But this data definitely warrants further investigation. However, this did result in the dropout of our low disperser, the mayfly species, from our RADseq dataset. All of our specimens for our high and intermediate disperser category barcoded to the right species, so we moved on to RADseq and collected SNP data for that. So for our results, for Annex junius, our high disperser, we did observe a significant relationship between FST, or genetic differentiation between subpopulations, and geographic distance. So essentially, with increasing geographic distance between our subpopulations or focal sampling areas, we see a higher degree of genetic differentiation. With our intermediate disperser, we did not observe a significant isolation by distance relationship, but we did generally observe a trend of increasing FST with increasing geographic distance between our focal sampling areas. We also ran a structure analysis, which is membership likelihood using Bayesian clustering. And so how to read this figure, essentially each box represents a focal sampling location, and each bar within the sampling location box represents an individual. The height of the bar within each box represents the likelihood that each individual is a member of a particular cluster, whether it be the blue or the orange cluster. So our southern sites are on the left side of the bar plots and northern are on the right. For Annex junius on the top, our high disperser, we observed 2 genetic clusters, one in the southern 2 sites and one in the northern 2 sites with a zone of admixture in the middle. For Nodonecta on the bottom, our intermediate disperser, we also observed 2 genetic clusters, but we see a little bit more of a random organization. We see 1 cluster at Indiana Dunes, Warren Dunes, and Ludington State Park, while we see another genetic cluster with Saugatuck and Sleeping Bear Dunes. And this is a little bit more difficult to resolve, especially provided that Nodonecta did not display a significant pattern of isolation by distance based on the Mantell test. But generally for both species, we see 2 genetic clusters along the coastline. And so specifically putting our Annex junius results, our high disperser results for structure along the coastline, it's interesting to visualize that we have a southern genetic cluster, a northern genetic cluster, and then a zone of admixture in the middle of those 2 genetic clusters. For both Annex junius and Nodonecta undulata, we see significant relationships between genetic diversity and latitude, where genetic diversity increases with increasing latitude. This is consistent with similar studies on organisms including stoneflies, amphibians, and fish, where genetic diversity has been observed to increase with increasing environmental heterogeneity. In the case of interdunal wetlands, environmental conditions may be more heterogeneous at greater latitudes. So on the left within extraneous, we see an increase in observed heterozygosity with increasing latitude. And on the right for Nodonecta undulata, we see an increase in private alleles with increasing latitude. So wrapping up our results, for our high disperser and extraneous, we see significant signatures of isolation by distance. For both species, we're estimated to have 2 genetic clusters along the coastline. And for both species, we also see significant increases in genetic diversity with increasing latitude. We also provide an interesting updated distribution of Canis mayflies, where we see one species, Canis amica, at the southern sites and Canis diminuta at our 2 northern sites. Lastly, this is the first genomic assessment of aquatic insects from interdunal wetland habitat. We provide next generation data sets for 2 aquatic insects and also provide de novo sequences that can be used for future studies of these non-model organisms. So interdunal wetlands are globally ranked as G2 habitats, meaning they're imperiled due to rarity, and state ranked as S2 based on vulnerability to extinction. Based on our results indicating structure in a highly dispersive aquatic insect, our dragonfly, we recommend that interdunal wetlands are managed as a contiguous coastal habitat with a focus on management that prevents any disruption to genetic connectivity. Our results also indicate that population genetic structure cannot be directly inferred based on dispersal ability, and this elucidates the idea that inference of structure may need to be on a species or even habitat-specific basis. Future directions for this research can include the incorporation of a low disperser into next-generation sequence data sets, the inclusion of more landscape variables, including dune aspect, wind direction, canopy cover, hydroperiod length, and we can potentially investigate local adaptation within insect species along the coastline. Lastly, a major shout out to all of my funding sources and sources of support over the years for this research. And then I'm happy to field any questions. Feel free to send them my way during the molecular ecology moderated discussion, or feel free to email me or contact me with any questions. Thanks so much.